Our goal today is to understand the experiments that led us to think about neurotransmitter being released in small packages or quantum, and the role that calcium plays in regulating that release of neurotransmitter. In the 1950s, recording from the frog neuromuscular junction, Fatt and Katz observed that in response to nerve stimulation produced a large amplitude excitatory postsynaptic potential, which could bring the muscle to threshold and cause a muscle action potential and contraction. In conditions of low calcium, however, the amplitude of this postsynaptic response was diminished greatly and did not have a continuous range of amplitudes, but rather clustered around a minimum amplitude size and multiples of that size, either doubles or perhaps triples of that amplitude size. Similarly, spontaneous events in the presence of calcium also showed this same amplitude, minimum amplitude, and fluctuations around it at a reasonable frequency. But in the absence of calcium, these little events called miniature excitatory postsynaptic potentials were much less frequent. The names of these can be confusing. The motor end plate is another name for the neuromuscular junction. So these very small events are called miniature excitatory postsynaptic potentials, or miniature end plate potentials, or sometimes just plain MEPs or minis. And these minis were quite curious and still remain so. But in the absence of calcium, there were fewer of them, but there appeared to be what we think of as a minimum size below which we do not see any amplitude fluctuations. So what role does calcium play in regulating the release of neurotransmitter under these kinds of conditions? Does calcium have a presynaptic role? Does it have a postsynaptic role? Blocking voltage-dependent sodium and potassium channels did not block the synaptic responses. The only thing that blocked them was complete removal of calcium, that is, the evoked responses. So Katz and Milady performed the following experiment, where they bathed the neuromuscular junction in calcium-free fluid, and they recorded both from the muscle and stimulated the nerve, and then they placed a pipette which contained calcium in it very, very close to these end plates or neuromuscular junction, and they could ionophorese or squirt out very small quantities of calcium locally to supply the need, and they altered which came first, the nerve stimulation or the calcium release. Under conditions when there was no calcium present, stimulation of the nerve produced very little to no postsynaptic response. Similarly, calcium by itself was insufficient to produce a postsynaptic response. If the nerve was stimulated first and calcium was added after that, again, no response was evoked in the muscle. Only when the calcium was present prior to the nerve stimulation was there a reliable postsynaptic response, indicating that the calcium was needed for some kind of an event at the nerve terminal involved with nerve stimulation in order to produce the postsynaptic response. And it's from this critical experiment that we understand that calcium's role is a presynaptic role as opposed to a postsynaptic role. And throughout all of this, if one brought another electrode with acetylcholine in it and puffed small amounts of acetylcholine onto the neuromuscular junction, one could record postsynaptic responses, irregardless of whether. So the amplitude of these miniature end plate potentials varied ever so slightly, and when the amplitudes were plotted, they formed a perfect Gaussian distribution. That's true in the absence of calcium and in the presence of calcium, and the larger evoked potentials also in the presence of calcium formed in very nice Gaussian distributions. But in low calcium, the miniature end plate potentials, or the evoked release, formed a very different kind of a distribution, one that was not cleanly Gaussian, but had peaks and troughs. So Del Castillo and Katz treated that in a stochastic manner, and they formed the quantal hypothesis of transmitter release. That is, that neurotransmitter was released in specific packages, and each package had an amplitude equal to one hump in the peak. So Del Castillo and Katz applied the known mathematical analysis known as binomial distribution, and they assumed that a single package of neurotransmitter, or a single quanta of neurotransmitter, produced a minimum amplitude of postsynaptic response that they called the quantal amplitude, and abbreviating that with the letter A. And they thought about the probability of release of this single quanta. Probability of getting success was P, and the probability of no success or failure was Q, or 1 minus P. The number of quanta at the nerve terminal was unknown, some little number n, and every time they stimulated the neuromuscular junction, that constituted a single trial, and you could control that experimentally for a total of capital N trials to produce a total number of releasable events. But on any one trial, the probability of releasing an unknown quantity X of quanta, that is, how many packages of neurotransmitter could be released, would be described by the binomial distribution, where the probability of releasing X, where X was a low number, 0, 1, 2, etc., was equal to the number of little n factorial times P raised to the X times Q raised to the n minus X over n minus X factorial times X factorial. Now, and if we do this capital N times, we run the experiment at large N number of times, the number of trials containing that will show that 1, 0, 2, 4, etc. packages or quanta were released will be the number of trials times the probability, P of sub X. So that gives us N sub X, and that's just the same equation with a capital N in front of it. And here we have 3 unknowns. We have little n, little p, and little q, and we can control capital N. So not quite a situation that's perfect for experimental control. So in the binomial statistics, P and Q, if you get one, you can get the other. They're simply related. But the little n is unknown, and the two variables n and p are difficult, if not impossible, to get experimentally. So Del Castillo and Katz needed a simplification. And it turns out that binomial statistics are related to Poisson statistics if one assumes that n and p, when multiplied by each other, are a theoretical value m. Under those kinds of conditions, that binomial equation for the number of trials producing a result of quanta little x can be simplified to this equation: n equals m to the x over x factorial times e to the minus m. And there, one only has 2 experimental variables, n and m, that need to be determined. So what is m actually going to be? Under these kinds of conditions, m is the mean number of successes per trial. That is the total number of quanta released over all trials divided by the total number of trials. So it's basically the average number of quanta released in any one experiment or experimental stimulation. It can also be calculated over an entire experiment by calculating the mean evoked potential amplitude and the mean minimum mini evoked potential amplitude, and dividing one by the other. And so we call that quantal content, or the average number of quanta released in any one particular presynaptic stimulus. Now, there's some assumptions that Poisson makes in order to have this simplification, and that critical is that each quanta be released independently, that no one trial is influenced by the previous trial, that is, there's no memory or hysteresis in the system, and that P approaches a very small number and N approaches a very large number, so that their product can become this new variable M. And under these conditions, variable M. And under these conditions, then Del Castillo and Katz could use the predictions of how many trials should produce 0 quanta released, 1 quanta released, 2 quanta released, 3 quanta, 4 quanta, etc., and predict mathematically what the distribution of amplitudes should be, and then match that to the experimental results. What I'm showing you actually isn't the data of Del Castillo and Katz. It's actually a later experiment done by Boyd and Martin where the fits were so perfect that they have become the figures of choice for textbooks ever since. But the process was the same in both sets of experiments. What we want to pay attention to is this column of what we call failures, the number of trials out of the total number of trials in which no responses were recorded. And this just shows you how the Gaussian distribution is applied to each of those peaks of the predicted amplitude in order to get the previous. So if we consider the number of failures where x is equal to 0 and substitute that into the previous equation, the denominator turns out to be equal to 1, so this simplifies even more, and the number of times one counts failures should be equal to the total number of trials times e raised to the minus m, which is a much simpler equation and can actually be used to solve for m. So inverting this equation, m is equal to the log of the total number of trials divided by the number of trials in which one had failures. So that's a very nice experimental way to determine m, the average number of quanta released in any one trial. But we also have this other way that m can be measured by taking the overall mean evoked response and dividing it by the overall mean miniature response. And this is what Del Castillo and Katz did. They measured m in both ways, and they plotted one versus the other. So the mean end plate potential divided by the mean miniature end plate potential is on the x-axis, and m calculated as log of n over n0 is on the y-axis, and you can see how their experimental data lies upon the unity line right in the middle, which is about as good an experimental result as one could ever expect from a set of experiments. Nailing the idea that a quanta of transmitter is somehow or another very essential for this release process, and that we have now this mathematical model of stochastic release that the neurotransmitter release is apparently obeying. That is, conditions in which the probability of release is relatively low, and the number of
So this is
possible
consistent
things that
with
can
the
be
idea
released.
that
Packages
the probability of
neurotransmitters
release is a
relatively
very
high.
low number, and the pool of elements to be released, or quanta to be released, is a relatively large number, and that together they produce this idea of quantal content. That is, how many release events occur. And notice in this whole analysis, the amplitude of a single event at the postsynaptic site, which is equal to A, really only enters in to this miniature mean amplitude. So that's really this minimum response postsynaptically. The whole postsynaptic response is being really used as a bioassay for the mechanism, for understanding the mechanism of presynaptic release. Now we do know at the neuromuscular junction that there is a high efficacy, a high probability of release. So the model doesn't perfectly fit the neuromuscular junction, but is much better at fitting in the central nervous system, where the probability of release is a very low number. And the real question becomes, experimentally or in biological terms, what does little n represent? Does it represent the number of vesicles? Does it represent the number of vesicles that are docked and primed? Does it represent the number of nerve terminals? Because in the central nervous system, multiple nerve terminals impinge on any one postsynaptic dendritic spine. And pre to postsynaptic connections are very seldom just one synapse each on each side, but are clearly multiples. So the physical interpretation of these are sometimes difficult to add to our whole conceptual understanding.